Good morning, everyone, and thank you very much, Paul, for the introduction. And again, I would like to thank the Marcus Wallenberg Foundation for this prize. It's a true honor to speak about parts of my career. So, So the main aim of the research in my team in Belgium is to support transition from fossil-based in a bio-based economy. And we believe that woody biomass is a very promising resource because many products that are nowadays made from oil can be made from woody biomass. And the reason is that woody biomass, all the fractions of woody biomass can be used in society. There is the fiber that can be converted to pulp for packaging and tissue and all kinds of new innovations. The cellulose and the hemicellulose polysaccharides can be depolymerized in a process called saccharification to primary sugars, and these primary sugars can be fermented to, for example, biofuels, but also building blocks for bioplastics, detergents, etc. And the lignin can nowadays also be catalytically fractionated into building blocks for the chemical industry. And lignin, as you already heard from Claire, is a main limiting factor if you want to convert wood into either pulp and either in a biorefinery into primary sugars. So there's quite some interest in reducing the amount of lignin in trees or in plants in general if they are used to improve digestibility. And this can be so either reducing lignin content is very important, but you can also change the lignin composition such that the lignin becomes more reactive to the chemicals and is more easily broken down. And this is an area of research where my group has been doing quite some research in. So to recapitulate, this is the monolignal biosynthetic pathway, and lignin is mainly composed of coniferyl and sinapyl alcohol and actually minor amounts of paracoumaryl alcohol. Thus, when these units are incorporated into the polymer, they are called H, G, and S units. The pathway has been very well described, and that means that we can overexpress or downregulate the genes in the pathway. And in this way, we can create plants that produce a lower amount of lignin or a different lignin composition. And so when these monomers are translocated over the plasma membrane and arrive in the cell wall, they will be oxidized by laccases and peroxidases into monolignal radicals, which will be combinatorially coupled into these standard lignin polymers which grow between the cellulose microfibrils in the cell wall. And they make that cell wall strong and hydrophobic such that plants can grow upward and transport water and solutes through the vascular system. Now, today I would like to give you 3 stories. One is how we moved from gene discovery in the model plant Arabidopsis up to trees grown in the field, trees with improved wood properties. A second example is how we can use gene editing using CRISPR-Cas to tweak enzyme activity. And in a third example, I would like to show you how we can make completely novel lignin polymers with different properties. So the first is about gene discovery. Actually more than a decade ago, we asked ourselves the question, are there any routes in that lignin biosynthetic pathway that are undiscovered? Are there any new genes playing a role potentially in bypass pathways? And that was a curious question in a way because we were looking for something that we didn't know that would exist. But you can do that in biology because you can make a co-expression analysis. It's a kind of guilt by association approach. It actually means that we are looking to genes that have an unknown role, an unknown function, and we investigate, we try to identify such genes that behave exactly the same as all the other genes in the lignin biosynthetic pathway. So without going into more detail, so we make a co-expression analysis, so we make a network of co-expressed genes. And we found that in subnetworks B and C, we found all the known lignin biosynthetic genes plus a few other ones. And so these are, of course, prime candidates to also play a role in lignin biosynthesis. And the way we study the role is by analyzing mutants. So at the left side here, we have the wild type and 2 mutants in the CZ gene. And then we made sections through the plant, through the stem, and we stain them with a lignin stain. And as you can see, the amount of the intensity of the lignin stain is much reduced. And there was a collaboration with Claire Halpin. And of course, because the lignin stain is reduced, we hypothesized that those plants make less lignin. And of course, we have to support that with wet chemistry data. That's what we did. So these plants, they have 30% less lignin. And then in collaboration with John Ralph, we found that the frequency of H units was dramatically increased. In wild-type plants, we only have 0.3% of H units. It's really a minor unit. In the mutant plants, the most abundant unit was the H unit, 44% all of a sudden. So if we knock out that gene, we have a less lignin and a strongly altered lignin composition. But these data do not actually show us where the gene plays a role in the pathway. And to investigate what the gene is actually doing and where it is positioned in the pathway, we use comparative metabolomics. It means that we extract metabolites from wild-type plants and from the mutant, and we separate that on UPLC in one direction based on retention time, and then in a second direction based on their mass over charge ratio. And so we can separate about 3,000 peaks from a single extract. And using mass spectrometry, we can fragment those metabolites, and based on the fragmentation pattern, we can puzzle the compound back together. So this analyzing the fragmentation pattern tells us the structure of the molecule. So if we do a comparative metabolite profiling of the wild type and the mutant, we saw that caffeoyl shikimate was a compound that massively accumulated in the mutants. And this rang a bell because caffeoyl shikimate was an already known pathway intermediate. So to discover the role of the enzyme, we heterologously produced the enzyme, and then we fed it with caffeoyl shikimate as a substrate. And using these enzyme assays, we saw that caffeoyl shikimate is converted into caffeic acid. So we discovered a new path, a new piece of the pathway. But it's an important piece because if you knock out the gene, the production of lignin is much reduced, and because we have 30% less lignin, and at least part is compensated, the flux goes into paracomyl alcohol, which is a very abundant monomer. Now, very interesting is that if you saccharify the stems of these plants, the saccharification is enormously increased. So here you see a time course experiment where we plotted against the percent of cellulose that is converted to glucose by an enzyme treatment of the biomass. In wild type, only 18% is converted in our mutants, in the knockout mutants. This is increased by fourfold. Almost all the cellulose got converted, and this without any biomass pretreatment that is usually used to open up the cell wall and destroy the lignin to a certain extent. That was very promising. So therefore, we translated that into poplar. And we achieved poplars, and this is greenhouse data, that have up to a quarter less lignin. And these trees, much to our surprise, did not have any yield penalty. So therefore, we took those trees further into the field. We propagated them. This is collaboration with the Ilvo station, and we flew over a drone. And so we have the wild type and 2 transgenic lines, each in 48 copies, and this divided in 6 blocks in the field, and this is to be able to do proper statistics. So if you look to the drone flight, there does not seem to be any yield penalty. This was a big surprise because I would really have put my hand in the fire that we would have a yield penalty, but there doesn't appear to be one. But of course you have to measure this. And so we harvested 3-year-old trees, did very strong statistics. We didn't have any yield penalty. And lignin measurements told us that there was a reduction in lignin content by 13%. It's less reduced as compared to the greenhouse, but that's something we always see. And that translated to a 90% increase in saccharification efficiency. So it means that upon an alkaline pretreatment, so it means that the biomass is more easily saccharifed because there is less lignin, cellulose microfibrils are more accessible to the enzymes. And together with Troy Runge's lab in Wisconsin in Madison, we performed kraft pulping experiments with the wild type and the 2 transgenic lines in 8 different conditions with ranging active alkali, sulfidity, and H factor. And then based on these data, and these are actually quite fresh data, models can be built and then you can ask questions to the models. So these data show you, or the model tells you that when you have conditions that with the wild type plant you reach a kappa number of 15, and the kappa number is the amount of lignin in the pulp after the pulping, that the transgenic lines will have a kappa number of 13, and at the same time they will have 1 to 2% point more crude pulp. So this is certainly interesting for the pulp and paper industry. So I've shown that gene discovery and very fundamental research in Arabidopsis can lead to very interesting and promising data for applications in biorefinery and in pulping. A second story I would like to tell is about genetic gene editing with a focus on another gene in the pathway. So cinnamyl-CoA reductase catalyzes an important step in the production of these monolignols. Using CRISPR-Cas, we can knock out the complete gene. We can make it dysfunctional. CRISPR-Cas is a system that you introduce in the cell. The system goes to the target gene, in this case CCR. It cuts the DNA split. The biological system will repair it, and sometimes it makes a mistake, and therefore the gene will be knocked out, will not be functional anymore. And so if you knock out the gene, your plants will be very small. They will saccharify very well, but they are too small, not interesting for applications. But it does not mean that CCR is not a good target for applications. And I will explain you why in the next slide. So our tree that we use is a hybrid tree. It's a combination between Populus alba and Tremula. Actually, all trees are hybrids, like humans. And so that means that we have one copy of the CCR gene from Tremula and one from alba. And the gene editing machinery has made, and the arrow apparently points wrongly, but a single base pair insertion in one copy, which makes this copy defective, and it makes an 8 nucleotide deletion in the other copy. And this makes also the gene defective, and that's why we have these very small plants. But we had also plants which have one insertion in one allele, making that allele defective, and a -3 nucleotide deletion in the other allele. And 3 nucleotides is the same as a codon. So that means that if you translate in silico this new edited allele into the protein sequence, you will create a CCR protein, but with 1 amino acid less, and also with 1 other amino acid, because the 3 nucleotide deletion was not completely in frame with the reading frame. Okay, but you do create a functional CCR protein, and those plants that are gene edited, they are normal in appearance, but they have reduced lignin, because if you peel off the bark, you have this typical red xylem coloration, which is a hallmark for CCR deficiency. The plants have 10% reduced lignin, and they have a significant improvement in saccharification efficiency without or with an acid or an alkaline pretreatment. And so these trees are now in the field in collaboration with Sweet Tree Technologies in Sweden here. A third example is how we can make completely novel lignin polymers that are not yet potentially found in nature. And this story also starts from our co-expression analysis. So we identified a new gene that appears to make a protein called CoZ that takes 6-hydroxyphenylvyl-CoA into scopoletin being a coumarin. And 6-hydroxyphenylvyl-CoA is derived from phenylvyl-CoA. It's a part of monolignol biosynthetic pathway with an enzyme called F6'H. So how did we discover the function of that gene? Again, we used comparative metabolite profiling. So we analyzed the metabolites that were increased in abundance and decreased. So in red, those that are increased. And using mass spectrometry, we tried to unravel as many structures of compounds that accumulated and disappeared. And by comparing the structures of the red and blue boxes there, we hypothesized that 6-hydroxyphenylbyl-CoA was converted to scopolamine, and this we could then actually show in an enzyme assay. Now, the neat thing for this talk is that scopolamine shares structural similarity with a coniferyl alcohol, so we potentially can use it to incorporate it into the lignin polymer. I'm going to give some more details here. So this is again the 2 steps from phenylbyl-CoA, a pathway intermediate in monolignol biosynthesis, towards scopolamine. And this is coniferyl alcohol. So where does it share structural similarity? Well, scopolamine and coniferyl alcohol, they have this para-hydroxy function. This is needed to create radicals, biolacases and peroxidases. The molecules have the 7-8 double bond, which generates the ether linkage in lignin, and ether linkages are the most frequent units in lignin. And scopolamine has this additional advantage that it has an extended conjugated system. So that means that if we can boost the abundance of that molecule in lignifying tissues, if it incorporates into the lignin, it will create ether linkages that are more susceptible to alkaline degradation. So our objective was to produce scopolamine, which is normally only present in roots. Objective is to make it in stems. And that's why we expressed F6'H and COSY, these 2 genes, under the control of a strong lignify, a promoter that lignify, but is expressed in lignifying cells. And analytical tools then showed that the plants really they produce more scopolamine, and also its glycoside scopolamine. And you can even see the incorporation of scopolamine in lignifying cells in Arabidopsis based on its fluorescence characteristics. So these plants, they do incorporate scopolamine in the lignin polymer, as demonstrated by NMR collaboration with John Ralph's lab. At the right panels, these are NMR spectra from the transgenic lines, and at the left of the wild type. And at the left bottom here, you see a difference spectrum. It subtracts those 2 spectra, and therefore you only see the differences. So the red contours reflect the scopolamine molecule incorporated or attached to lignin, because they are the same contours as NMR spectrum from pure scopolamine. Now, finally, upon an alkaline pretreatment of this biomass of Arabidopsis, you can see we got a 40% increase in saccharification efficiency. Again, this was very interesting because the plants did not have yield penalty. Therefore, again, we translated that research into poplar. We have trees now growing in the field that produce scopolamine, and so far they're growing well. And if you want to know more about these trees, we have a poster from Nette de Ridder hanging on the hall here. And we have also another poster by Chantal Anders, who is following up another gene derived from this co-expression analysis. So, in conclusion, we can make trees that are better for industry and for the environment. So, instead of making trees that have a higher biomass yield, we make trees that have a higher yield of the product you can make from wood. So we can save chemicals, we can save energy, so we can make more product from a plantation. And that should, I hope, reduce pressure on harvesting native forests and the associated biodiversity. And in this context, I think it's important also to say that genetic engineering and gene editing, they are not alternatives to classical breeding. Genetic engineering and gene editing are tools that create extra genetic diversity and phenotypic diversity. And these tools need to be integrated with conventional breeding to make trees that are suited for the pulp and the biorefineries. And I would like to close, of course, by saying that I did not do this work alone. The publications that I discussed are listed here. Also, the groups that have been very important in that research, even if I did not show all the data that these produced. This is my group in Belgium, so you will recognize Nette and Chantal, who will be presenting a poster here, and Tatjana Mota, who has been much involved, or well, responsible for the work on the poplar field trials that is not published yet. Thank you very much for your attention.